All right, so thus far we've talked about phylogenies, we've talked about taxonomy, and now we're going to talk about systematics. We're going to talk about how you create those cladistic trees. We began talking about cladistic trees last time. I think you guys came up with the brilliant solution of talking about like trying to figure out how a quokka is related to other organisms. So thanks for the and a cardinal. Um, Now we're going to talk about a little bit more in depth on how to make those trees more refined. So they're not just sort of our random shot in the dark hypotheses of how these are related, but they've got some evidence to back them up. Most of systematics. So systematics is all about describing how these organisms are related to each other, is based on morphology. Species are usually grouped together by shared characteristics. Take a look at these guys. What do they look like? They're dolphins, and they totally are dolphins, but they look different than normal dolphins. You guys have a conception of a dolphin in your head, right? How do these guys look different than normal dolphins? Yeah, right? They got super thin. These are called rostrums. Super thin rostrums. Look at their eyes. What do you think of their eyes? Teeny tiny little eyes. Big, wide flippers up top, but they're very thin tails at the back. These guys are all adapted to a different environment than other dolphins are adapted to. This is the Yangtze River dolphin. Functionally extinct as of now. It's technically went extinct. The last time they did a survey of the Yangtze, they found in 2006 there were no more river dolphins. This is the first megafaunal organism in not recorded history, in our modern history that's gone extinct due to human influence. These guys all make up one group of dolphins because they have these shared characteristics. Make sense? Then you can further subdivide them. You've got lighter colored ones, dark colored ones, you got pink ones. Had pink ones. You group a species together based on their shared characteristics. Geography can also be important to this whole thing. Even though these organisms may look alike, based on where they live, they would be a separate breeding population. Most organisms don't migrate over huge spans, except birds, really. Okay, guys, what is this? It's a cat, exactly. You're like, that's a cat. I know this cat. This is a very specific subspecies of cat called the Manx. The Manx cat is missing its tail. It's got much longer, more powerful hind legs. The Manx cat evolved on the island of Man, just off the coast of England. This subspecies of cat is well known for basically it hops. It looks like a little rabbit. The Manx breeds within that Manx population because it's on an island. It doesn't swim well. It can't get to the mainland. It's becoming its own unique species. So geography is important to speciation and defining what a species is. We can start classifying organisms by how they might be related to other organisms. This is sort of a classic trilobite example. We can use the fossil record to say, these guys, again, morphology, look a lot like those guys. You start to find organisms that are almost identical to contemporaries. If you think of the Glyptodon, which you don't know what it is, think of do you know what an armadillo is? Okay, imagine an armadillo the size of a VW bug. So that's a Glyptodon, which is basically a giant armored rat. The Glyptodon looks just like the armadillo. It does things similar to the armadillo. They are related. It's almost a direct line of lineage from the Glyptodon to the armadillo. And you can create these systematic charts based on what's related to what. Sometimes we just start to group these organisms together by morphology. Homoplasy causes trouble with that. If you say everything that has wings is in the same group, you'd be wrong. You got bats, you got birds, you got pteranodons, you got all sorts of different things that can fly. You got insects. Even though they have the same basic structure, the wing, it might be adapted from different parts. If we look at a bat's wing, for instance, you're looking at, here's the, this is a good example. You got the elbow, it comes out, and then you got digits, your fingers, in which all of the webbing occurs. Versus a bird. In a bird, you've got a wing is basically one long digit, and all the others are supporting it. It doesn't fan out as much. Homoplasy results in what's called convergent evolution, in which similar structures, analogous structures, are going to arise independently in two different lineages. The shark, the ichthyosaur, and the dolphin all look similar. Why? You got a fish, a reptile, and a mammal. They all have the same basic shape. It's called thuniform. Why do they all have the same basic shape? Right, they all live in the same area. They live in water. That just happens to be the best shape for being agile and fast in water. So that's conversion. We see it with organisms that swim. We see it with organisms that fly. Turns out the best way to fly is to have wings. So they, sorry, I just imagine millions of years in the future, a squid being able to fly with jet propulsion. But the falcon, the bat, the pteranodon, they all have different, they all have wings, but different ways that the wings evolved. The armadillo, the giant pangolin, the giant anteater, the spiny anteater, they all look similar. How do they all look? What do they all have in common? Long faces. They all have sharp claws at the front. They all have what look like armored or protected bodies. The reason for this is, of course, they live in similar environments and hunt similar prey. They all hunt burrowing insects, termites and ants. To get termites and ants, it's super useful to have a long, sticky tongue. To have a long, sticky tongue, it's good to have a long, sticky, or a long snout in which that tongue is anchored. They have strong claws in order to dig, because you want to dig down into the mound. And probably one that's the most common cited example is the human eye and the octopus eye. The human eye and the octopus eye evolved independently, but are structurally almost identical. Different genes get you there. Our eyes evolved in a similar pathway to the octopus eye. It focuses in the same way we do. Now, we'd say normally, well, why does that work? Because we live in the same environment, but we don't. Octopi live in the water. Octopi actually can, I'm sorry, octopodes is, I guess, the correct term. I keep getting that wrong. I always call them octopi, and it turns out it's either octopuses or octopode, because it's a Greek root. Now you know. The octopus uses its eyesight in the same way we do to hunt, and it uses it in order to camouflage itself. So it evolved the same basic. It's got eyelids, it's got a cornea, it's got a pupil, it's got an iris. It focuses its the light from outside to the back of the eye, where it's interpreted as an image. A big difference here you can see is that the octopus eye has a lot more areas for optic nerves and for blood to flow into the eye, but that's neither here nor there. It's the same basic structure. It just evolved in a completely different lineage. Evolution. So we've got morphology we can use, we've got environment or location we can use. We can also use what's called molecular systematics, and this is where it gets messy. And I think you guys saw this in your lab when you were counting the differences between an amino acid sequence in horses and in donkeys and in whales. Yeah, that took like forever to count those, didn't it? Right. And you're just like, I'm done. Nope, not counting it. The good news is you guys were doing by hand what computers now do for us very, very quickly. Basically, you're going to be given, here's 10 different organisms, right? These 10 different organisms all have and this is a nucleotide sequence. They're different lengths. So what we've done is we're trying to, in molecular systematics, line those up in the most simple way that we can. Maybe add some spaces in. So what I've done in this next section is align the data. We've introduced gaps to make everything line up about right. So if you look here, all the first one starts with A, then all the second C, then G, then T, then C. This section, these first 5 nucleotides in B, don't tell us any differences between these organisms. They are not useful. Does that make sense? Then we see a gap. And then was it GTA all the way down. So this gap and these GTAs tell you that these 4 are closely related, and then these 6 are also related. You've got a branch. Down here, you see the same thing happening with brown, then pink, green, teal, red, blue, and purple. Now the issue is, you, the scientists, need to figure out what is the easiest way that this could have happened. Obviously, we see a distinction between, for the yellows, between the first 4 and the last 6. But what possible way can we draw this tree in which all of this occurs? And it turns out the most parsimonious, the simplest, is to say the biggest change, here we got 6 of them have a blank. There's an addition of T in these 3. So blue has, 7 of them, and then 3 are missing it. There was a deletion there, or an addition, we don't know. So we have our first big split between number 10 and basically all the rest. Then we have a split between green and yellow. Here. Oh, yellow and pink are together because they always travel, it's the same 4. So we got green and yellow and pink. Just looking at the green ones, we've got a split between that teal and the brown. And then some sort of back mutation, a reverse mutation between 8 and 9. In with the yellows and purples, so this group down here, which is 1, 2, 3, and 4, you see a split between red and purple. So you can say that's where they split apart. That's the simplest way of making this. Is this the only way? No. You could have come up with other ones, but this is probably the simplest way for it to have happened. So that's molecular alignment. It's what you guys were working on with your lab. When you run these out using various government databases and some analysis software, you're going to get confidence intervals. You're going to probably build about 100 of these. You're not going to, the computer is. And that's going to give you a percentage of, this is the likelihood of this one, this is the likelihood of that one. It doesn't mean it's right, it just means that it's likely. So that's systematics, figuring out how these organisms are related to each other. You could do it with morphology, which is, if we do it in class, I'm going to have you do morphology. We could do it with where do they live. We could do it with what's in their DNA. We use systematics to explore cladistics, or creating groups. Remember, clade means group. So we're going to classify organisms based on their evolutionary relationships to each other. This cladogram should look familiar. We've got 1, 2, 3, a few different animals, and then we've broken those animals up into different groups, trying to figure out what's the outgroup, which one's the most different. In this case, the most different is a turtle. And then all the figuring out how the rest are different from each other based on morphology. So if we take a look at this group, the turtle's the obvious outlier. It's a reptile. It's got slimy skin, no hair. From there, we have a break. And all of the rest of the organisms have hair. Every single one of these. So we put hair there because all of them have hair. Then we knock the horse off because all of the rest of them are carnivorous. Then we knock the, let's say, retractable claws. The wolf has no retractable claws. The leopard and the cat do. And surprisingly, what separates the leopard and the domestic cat is the domestic cat purrs. The leopard doesn't. And that's about all that separates a leopard from a domestic cat. And size. Oh, they do? Well, then that chart's wrong. So there we go. We revise our chart. We come up with something different. So cladistics compares differences in a single trait. I'm sorry. Cladistics compares two organisms with differences in a single trait, and that's trait is called a character. One type is called a simplesiomorphy. This is where a character is shared by two or more organisms. This is a shared primitive trait in this particular group, or in the last group. Hair would be the simplesiomorphy because so many of them shared hair. Simplesiomorphs are when they're shared by a whole bunch of the organisms. It's also a great Scrabble word. If you can work simplesiomorph into a conversation, like, you win science. The flip side are the synapomorphs. Synapomorphy is where it's shared by the most recent common ancestor. So here, it's not shared by a bunch of them, it's only shared by a few. So simplesiomorphs are very common among a group. Synapomorphs are what separate two groups. It's the one trait that separates two. It's also called a shared derived trait, but simply because I like big words, synapomorph and simplesiomorph are the best way to go in the class. Cool. We got in-groups and out-groups. It's just like high school. The in-groups are the ones that you're interested in. That's what you want to understand. The out-groups, they're the ones that have you figure have diverged before the species in the in-groups. Usually these trees are rooted by an out-group, the one that you are comparing it to that is the most different from all the rest. In the last example we looked at, the turtle is the out-group. It was the one that's most different. It's the one you can compare all the rest to. So you are interested in in-groups, and you use an out-group to compare to the in-groups. You knew you were cool in high school because somebody else was less cool than you. Does that make sense? You guys know that's you measure yourself against other people. You have to measure these trees against other organisms. So, how to make a cladogram. And I guarantee you, no matter what version of the assessment you're taking, you are going to have to build a cladogram. You're going to choose the species you're interested in studying. You got to figure out what characteristics you're going to use, determine what traits are primitive and what are derived. Then you're going to build the cladogram with species at the tips and characters at the nodes. And then you figure out what's the simplest possible explanation. So we did this in class before. You did this for your lab. Basically, you're going to build a quick cladogram based on, I'll tell you what the assessment, I'll just give you an assortment of different organisms that you may or may not know anything about. And all I care about is your reasoning and how you build it. So for, let's say, this, we've got several different organisms. I'm just going to do basically the same thing we just did. Turtle as a reptile, horse, wolf, cheetah, cat. Now, I'm not taking the time to go through all the reasoning, but you could have listed out all the organisms and said, what are their characteristics? What are we interested in? Cheetah and cat. And you're going to start putting pluses and minuses all throughout. So we're going to create a table with different characteristics for these different organisms. We're going to start out listing the organisms. Turtle, horse, wolf, cheetah, cat, all along the top. Down the side, we're going to put all of our different characters. Hare, carnivore. Let's see. Retractable claws. And then what is the difference between a cheetah and a cat? What's different between those two? Say it again. Purring? Oh, yeah, we talked about purring, even though it may not work so well. Um, sure, size. Size isn't a good distinction between all of them, but we can do size. Let's do spots. Don't cats have spots? Some cats have spots. Not all cats. Do all cats have spots? Cheetah print. Cheetah print. I'll write that. Cheetah print. Cheetah print. Okay. So we've got. Do turtles have hair? No. So they get a negative. Are they a carnivore? No. They really love strawberries. Surprisingly. Do they have retractable claws? Do they have cheetah print? Horses have hair. Wolves have hair. Cheetahs have hair. Cats have hair. Horses are not carnivores, but wolves, cheetahs, and cats are. Retractable claws. Horses don't have it. Wolves don't have it, but cheetahs and cats do. Cheetah print. Horses don't have it. Wolves don't have it. Cheetahs do, and cats don't. So we can go through and we can say, here are the differences between these organisms. We've got hair is missing in all of these. Our turtles are outgroup. So we're going to put hair right in between the turtle and the horse. Between the horse and the wolf, we then get carnivore because we've got the symplesiomorph. Now we're looking at the synapomorph. So the synapomorph here is carnivore. Then we've got retractable claws. And then we've got cheetah print. Oop, that's really spelling it wrong. So we've created a tree here that has not only how we believe these organisms might be related to each other, but also what traits separate them. And that's going to be important. What I normally see is people get the, I'm going to set up a tree right, but they forget to put the traits. You got to explain your reasoning. Nobody will get this answer wrong if you have some reasoning, even if your reasoning is stupid. And you guys know some of your reasoning is not going to be the best. It's going to be like, I don't know, cheetahs like bubblegum. I don't care. Whatever you want to put, as long as you explain your reasoning in a chart like this, you'll be fine. We always go for the simplest possible chart. It's the principle of parsimony. It's parsimonious. That is, it is the simplest possible answer. The preferred hypothesis is the one that is where is the simplest one for all characters in their states. This is also known as razor. The best hypothesis is the one that makes the fewest assumptions. Okay, so go for the simplest solution. Don't try and figure out a super complex way that this could have possibly happened just to make your original idea right. Always go for the simplest solution. If you hear hoofbeats, what do you think is coming? A horse. Is it possible it's a herd of zebra? Yeah, it's possible. Is it likely? No. It's probably a horse. I got two stories about this. One, the exception to the horse-zebra rule is in apparently Maryland, because there's a herd of zebra that's running loose that they haven't been able to catch. Not kidding. Somewhere in Maryland is a herd of zebra running loose. So be on the lookout. Look, there was a zebra farm, and they escaped from a zebra farm. I was, second story about Occam's Razor is, I was driving back to Charlottesville late one night. I was out with friends in Fredericksburg, and I was driving out to Charlottesville, and it was probably 1 a.m. Not the best time to drive. I wasn't very alert at that point. I'm driving along in the fog, and I look out the window, and I see in front of me a llama. I know, right? And I was like, you know what? I'm really tired. I'm probably just hallucinating. That's the simplest solution. There's it's a shadow I see in front of me. It's probably not, and it's not a llama, right? It's just the simplest possible solution is I'm tired and seeing things. And then I almost hit a llama in the middle of the road at 1 a.m. in Charlottesville. So I hit a llama. I didn't hit a llama. I almost hit a llama because it was one of those like, oh, I'm seeing things. No, that's a llama. Hit the brakes hard. And then I was really awake. Apparently, there was a llama and alpaca farm on the road in Charlottesville. I didn't know. The simplest solution to me was I'm seeing things. But if I would have known there happened to be a llama farm right there, it would have made more sense to me, and that would have been the simplest solution, which it turned out to be. There's a house that has emus. Look, there's a lot of my friend. Um, he we took very different paths in life. He ended up having a goat that was living with him. It was weird. So we can certainly figure out a parsimonious way of creating a cladogram based on morphology, but it's going to be heavily biased based on the researcher, what you think is important, what forms you think are important. So instead, modern taxonomy tends to rely on molecular sequences. Just like we did before, if you looked at these sequences, you can see some areas where they're exactly the same all the way down, some areas where they're slightly different. There's a C in there, there's a C in there, and so on. We use the differences to figure out how these things are related to each other. This only works because genes change over time, right? That's been like the whole course so far. Alleles change one generation to the next. Now, some alleles change much slower than other alleles. Some genes don't change very much. What kind of gene do you think would not change very often? I know, that's kind of a hard question. You got some genes that change like every generation. Some genes don't change at all over time because they're selected at remember, most changes will lead to a deleterious form. They reduce functionality. So what do you think won't change very often? Right, the traits that are vital to live in, the absolutely vital ones. Things like, how do you make a protein? Your ribosomes, those structures that build proteins, the DNA for those doesn't change often. It changes extraordinarily slowly. We've got different groups here. We've got fibrinopeptides, which, number of amino acid substitutions per 100,000, per 1,000 residues. You can see a whole bunch of them right off the bat. They separate out mammals. Hemoglobin, it's a fairly steady rate of change. Cytochrome C takes a long time because cytochrome C is useful. Do you guys remember what was cytochrome C useful for from your lab? Did you have to actually figure out what it did? No, that's BRCA-C, but close. No, it's not really. It's mitochondrial. It's involved in the electron transport. So if you can't transport, if you're not producing ATP efficiently, you're not going to survive long. So that's a very, very slow changing gene. We can use genes that are very, very slow to separate out U from yeast from bacteria. Not bacteria, U from yeast from amoeba. We would use something like fibrinopeptides, though, that change very, very quickly to separate out the mammals because they've diverged much more recently. So you got to pick the right gene to look at. So first thing you need to figure out when you're figuring out what gene to look at is about how long ago did you think that these organisms split off from each other? How do you think we could figure that out? How do you get a rough idea of how long ago we split off of the great apes? Yeah. Carbon dating is one way. You could totally use radioisotope dating. In order to do that, you need fossils or something from that time period to say, you know what, here's where a split likely happened. So yeah, radioisotope dating would be fine. We could do some other relative ways we'll talk about today, in the next lecture, I mean, to figure it out. So you figure out roughly how long ago these guys diverged, and then from that you could say, cool, then we're going to use this gene or that gene. Because like I said, some genes change very slowly compared to others. The genes that are involved in vital processes like ribosomes change extraordinarily slowly. The genes that, or the sequences that are, say, neutral mutations, neutral changes like in introns, those are going to change so rapidly we can use them to actually distinguish between people. You guys heard of genetic fingerprinting? in crime shows and everything. The genes that we look for, the sequences we look for there, are in non-coding regions because they have no impact whatsoever on fitness. So they're going to change very rapidly. There will be a difference between you and your siblings with genetic fingerprinting. If you tried to look at differences between you and your siblings with, I don't know, mitochondrial DNA, ribosomal DNA, you're not going to see almost any of them. There's going to be no difference between you and your siblings. Some are slower than others. Some very, very quick, like I said, the introns, they will change because they're not impacting fitness. Okay, so that is how you make a cladistic tree. But some things from molecular biology will muck it up. One is horizontal gene transfer, and we're going to talk a lot about horizontal gene transfer in the next several lectures. Horizontal gene transfer occurs when one species moves its DNA to another species. There's no mating involved. There's no another generation. You are simply getting genes from an entirely different species, and it happens to you, like literally to you. You guys have, I assume, at this point in your lives, all caught the flu at some point, right? No, never caught the flu. If you've never caught the flu, maybe you're genetically pure. The rest of us, if you've caught the flu at some point in your life, your body will actually incorporate some of the flu's DNA into your own genome so that you can fight the flu off again in the future. But you're taking a gene from the flu and incorporating it into your genes. It happens a lot more often. That's an extreme example. A lot more often among bacteria. If you get two bacteria together, they hang out close to each other. They will literally trade DNA back and forth. They're separate species entirely, but they can swap DNA. So if this one has some cool proteins on its outer membrane, the other one can pick those up. It's also why you get a lot of antibiotic resistance when you get two bacteria together. They share the traits that are most beneficial. As you can imagine, that horizontal gene transfer mucks up molecular phylogenies. It's likely, very likely, that the earliest life was not like we like to think of it as a tree where you have one root and then it grows out from that into this big, broad pattern. Instead, there was very likely a whole lot of single-celled organisms that were passing genes back and forth very, very readily. So you get this sort of web or bush at the bottom, and then you get a group of organisms that were going to go on to become proto-eukaryotes or whatever, and then they spread out from there. There's a lot of gene transfer. So don't think of it as a lot of individual species evolving. Think of it more as a community evolving together. As I mentioned previously, a lot of non-coding human DNA is actually attributed not to human genes, but to viral genes. Most of the Y chromosome, so guys, this is your Y chromosome, is made up of viral DNA. There's very, very few actual human encoding genes on it. It's a lot of just leftover genetic crap. So we tend to think of the web of life like this, where it's a single root with a whole bunch of branches. It's likely it was a whole bunch of different organisms coming together, spreading their genes back and forth, that eventually gave rise to more advanced forms. And then even later, you'll notice that archaeans and bacteria worked together, and they ended up passing their genes on to eukaryotes. Actually, they evolved into eukaryotes. You may have had a bacteria that got taken in by another cell to become a mitochondrion, or a plastid, a chloroplast, for instance. We're going to see a lot of horizontal gene transfer early in Earth's history.